Good morning, everybody. Thank you for attending the third webinar of the HealthFerm project. In this case, today we will talk about innovative pulse and cereal-based food fermentations for human health and sustainable diets on health aspects of plant-based fermented foods. And Professor Christophe Courtin, Professor Christine Verbeke and Professor Marjuka Kolomeinen will talk about health aspects of plant-based fermented foods, a point of view from the HealthFerm project. We have, as I already said, three presentations. Christophe Courtin will talk about plant-based food fermentation and why does it change the food and what should future research focus on. Christine Verbeke will talk about expectations on the effects of plant-based fermented foods, potential mechanisms, and Marjuka Kolomeinen will talk about the evidence that's yet available of the health effects of plant-based fermented foods. I do have some household rules for today. The presentations will follow each other without interruption. Participants can pose their questions in the question box. See the menu questions in the webinar, and please address the question to the presenter. And at the end of the webinar, collected questions will be addressed, and presenters will answer most urgent questions. We will pick out the questions with the help of Yamina Bond at the background, and question them to the presenters. And if your question is not answered right now, then an answer will follow in written as soon as possible. That having said, I introduce Christophe Courtin. He's a full professor of biochemistry, food biochemistry at the Laboratory of Food Chemistry and Biochemistry, and he's vice dean at the Catholic University in Leuven in Belgium, and his research focuses on cereal constituents with a focus on carbohydrates and dietary fiber, the enzymes that degrade them, and fermentation in cereal processes. He will very shortly introduce the HealthFerm project as such, and then go into the details with respect to the first presentation. Christophe, the floor is yours.
Thank you, Jan, for the introduction. Can you see my screen saying the HealthFerm project?
I think not. Veronica, can you change the presenter mode?
That has changed, so that should be fine.
Okay.
So it should be okay now. I just would like a cue from somebody that it's fine. Before starting. It's fine, it seems. Okay, perfect. Then I shall start. And again, very much welcome also from my end to this webinar. I'm Christophe Courtin. I'm the coordinator of the HealthFerm project. And I would like to spend a couple of minutes to present the project to you for those that have not familiarized themselves with it. So HealthFerm is a Horizon Europe funded project, a research and innovation action project. And in that project, we have gathered 23 partners from 11 countries, and a project that runs for 4 years. You can see the logos of all the different partners on the lower left-hand corner. As you can see, we have very nice arrangements of partners in the project. The budget for this is 13 million euros coming from EU on the one hand, but also from the Swiss government, because we have 2 Swiss partners also on the project. And the aim of the project is actually going into a transition from traditional fermented foods into sustainable plant-based fermented foods, with a focus on health benefits and with a focus on designing the microbial consortia that we need to ferment. So not so much spontaneous fermentations anymore, but more going into plant-based, into rationally designed fermentation processes, animal-based products going into plant-based products for a number of reasons. And all of that, of course, evidence-based. So science is at the basis of a research and innovation action. Overall, the focal points, if you look at the slide, so we have microbial resources, of course, very important. We have fermentation technology as one of the focal points. We have human health and microbiome, and we have consumer behavior. Those are the 4 big work packages that we're working on. If we focus more on health, what we try to do there is to look at how to disentangle the effect of food fermentation microbiomes, fermented grain-based foods, human health, and the human gut microbiome. So to see how these all interact in fermented foods that we consume to affect health. And that is something we believe is quite of interest because there's relatively little information available on plant-based fermented foods. That being said, I would like to continue with my presentation of today, which is in short called plant-based food fermentations. And this is a presentation that was made by Yamina de Bond, who is also very strongly involved as a scientific coordinator of HealthFerm, and then adapted by myself. In the lower right-hand corner, you also see the logo of Microbes for Sustainable Foods, which is actually a consortium of different European projects and one cost action in the area of food fermentations. So it's good to keep that name also in mind. With that, a small overview of my presentation. So different types of fermented foods, why food fermentations, what does fermentation do to foods, and then also future research very shortly. Now, if we look at the area of food fermentations, very often we come across the name microbial foods. And it's good to realize that if we talk about microbial foods, we're actually talking about 3 different types of fermentation that are present. On the one hand, we have biomass fermentation, where we produce microbial biomass, then we process that biomass into a food product. And one example would be corn, for example, which is probably known to you. We also have precision fermentation, of which we hear a lot these days. There we have a microorganism that we engineer and make sure that it produces a specific component. That can be an enzyme, that can be milk proteins, that can be something else. Then those proteins are typically purified and then transformed into a food also. What I would like to talk to you about today is more food fermentation. So the traditional concept where we inoculate raw materials with specific microorganisms, and we have a fermentation process going on, leading to a fermented food or a microbial food. So that is the focus of HealthFerm. We are not dealing that much with, or not dealing with microbial biomass. We're not working with precision fermentation. We're looking into food fermentations. If you look at fermented food products, then we have to make a distinction between fermented foods that have their live microorganisms intact at the end stage of the product. And then you can think about things like yogurts, miso, tempeh, kimchi, kombuchas that have not been pasteurized. On the other hand, we have fermented foods, of course, where the microorganisms have been killed off at the end of the production process. Let's think about sourdough bread or regular bread. It can be beer. Most beers are pasteurized after the production process. Fermented foods, and then of course some foods that are non-fermented, which to some might seem fermented or can be considered as fermented, but they are not. Acidified cottage cheeses, for example, fresh sausages, these are non-fermented. So it's good to keep that in mind. And as a rule of thumb or as a definition, I think we can look at the definition that was given by ISAP and Maria Marco in a very nice review, where they wrote that fermented foods and beverages are foods made through desired microbial fermentation, microbial growth, and enzymatic conversion of food components. Yeah. And so whether or not at the end microorganisms are present in the food, that is not part of that definition. Now, fermentation is something that has been happening for a very long time, and today up to 40% of human diets can contain fermented foods. Yeah. And on the left-hand side, you see plant-derived fermented foods. On the right-hand side, animal-based, so dairy products, meat products, seafood products. On the left-hand side, you see legume fermentations, fruit fermentations, grain fermentations, and so on. So quite a large spectrum of foods, up to 5,000 and more, are being produced on a daily basis around the globe and consumed. Now, what microorganisms are used? Well, it's bacteria, it's fungi, or it's yeast. Bacteria like lactobacilli, which we all know, Streptococcus, propionibacteria. If you look at fungi, it can be Aspergillus, like Aspergillus oryzae, very well known in koji, Penicillium rhizopus. We can also have yeast, of course, Saccharomyces being very well known, Saccharomyces cerevisiae, Pasteurianus, but also Clavibromices and others. And these different microorganisms will produce different fermentation metabolites. In this slide, you see glucose as a starting material for fermentation. You see ATP production, one of the reasons why microorganisms will ferment to produce ATP. And then you see that lactobacilli, for example, will typically produce lactic acid. Saccharomyces, ethanol, and carbon dioxide. Azotobacter and Escherichia will lead more to acetic acid. Propionibacteria, propionic acid, but also acidic acid and carbon dioxide. And then Clostridium, the much coveted butyric acid, for example. And so there's, depending on the choice that we make of microorganisms, we will get different metabolites and we will get different fermented products for sure. Yeah. This slide I wanted to include because it is a very nice, how do I make it, table made by Michael Gensler at the University of Alberta, and he calls it a kind of periodic table of fermented foods. And what you see there with the different color codes is the different microorganisms that are present in specific fermentations to end up with, for example, kombucha or water kefir. And so you see the complexity of the fermented food space. It's not a single organism, it's not a single starting material. We really have a very large diversity, which makes it very interesting, of course, as a research topic, but also for human food production. Now, if you look at the microorganisms that are used for fermentation, we in the old days, millennia ago, spontaneous fermentations were the only thing that happened. Then we went into backslopping, where spontaneous fermentations were reused and fermenting material could be sustained for a longer period of time. Today, we work a lot with starter cultures, of course. We have companies in the project also that are focused on production of starter cultures, where you have isolated microbial strains. You put them singly or in duo or in trio in a starter culture, and you add that to your fermentation in a very controlled manner. The next step, of course, is genetic improvement of starter cultures, where you can modify microorganisms to do more what you would like them to do. And then, of course, the next step is microbiome engineering, where you're going to make sure that you have the different microorganisms put together that will form the perfect consortium to do what you want them to do. Yeah. So that is a next step where you have much better control of the fermentation process and of the microbiome that you have. Okay, so that ends the first part of my presentation, so the different types of fermentations and fermented foods. And now I would like to go into why do we ferment foods and why are we looking in HealthFerm at food fermentations. And it is clear that in the old days, fermentation was mostly done for food preservation. And still today, that's an important aspect. But we look more and more at fermented foods also in terms of sustainability of delicious foods that we want to create with specific flavor profiles. We look into safer foods. Yeah, we can avoid some problems when using microbial fermentations. but also very important, we look at healthiness of the fermented foods. And that is a big part of what we want to do in the HealthFerm project. So sustainability, there, of course, we all know that the way we are producing food today might not be the most sustainable way. We are producing a lot of a high carbon footprint with our agricultural practices, with meat production and so on. Fertilizers are not optimally used. We are affecting biosphere integrity. So there's a lot of different aspects why we must try to make food production more sustainable. And microbial foods, be it biomass, be it precision fermentation, be it food fermentations, they can help to make our food production system more sustainable. I think that is an opinion of a lot of people. It's also the opinion of EU, World Economic Forum, and others, that we should go there for more sustainability. If we think more specifically for food fermentations, these are important or could be important in our transition from more animal-based products to plant-based products. Because if you look at the numbers and the carbon footprint that we are producing, the realization is that if we turn more to plant-rich diets, and I'm not saying diets without animal produce, that's not what I'm mentioning, it's more plant-rich diets, then you see that that change or that transition would have the largest impact on our carbon footprint in the food production system. And so that is where we want to go. The question is, what do we need to have more and better plant-rich diets? One of the difficulties, or some of the difficulties, is that plant-based materials are very often a bit more recalcitrant, a bit less functional than when you look into animal products. Animal proteins, for example, we all know that we can whip egg white, for example, to produce cakes and others, very functional proteins. Well, these things are typically more difficult when looking at plant proteins. We have a lower solubility of plant proteins, for example. We have a poorer techno-functional properties of those ingredients, but we can also have off flavors. We can have issues with food structure and texture. Very often mineral bioavailability is lower because of the presence of phytate that chelates minerals. Vitamin B12 is an issue that we always have to be careful of, other anti-nutritional factors, and low protein digestibility. So we have a lot of challenges when transitioning from animal-based products into more plant-based products. But we believe in the HealthFerm project that fermentation technology has the potential to solve several of those issues. So improve functional properties, affect the bioavailability of minerals, increase protein digestibility so that there's better use of those proteins when consumed. And that is actually what we are focusing on in the HealthFerm project. Safety, of course, is also a big issue, and there food fermentations can help to prevent the growth of pathogens in your food materials, can also reduce the problem of mycotoxins by mycotoxins binding to, for example, cell walls of yeasts. We can have reduction of phytates, increasing mineral bioavailability. So there's a lot of reasons, also from a safety point of view, to look into food fermentations. That has happened before, for sure, but we need to go further in that. And then we come, of course, to the aspect of health. Yeah? If you read magazines and if you hear people talk, then it's assumed that food fermentations are healthy per se, that it will increase the health profile of the food product. Now the question is, are we very sure of that? Because if we look into the literature and the scientific evidence that exists, we see that there is indeed scientific evidence for the health beneficial effects of dairy products, for example, yogurts and so on. there is much less evidence available on the health benefits of plant-based fermented foods. So there, if you look into the studies, you find things for sure, but very often the evidence is limited in what we see. Right. If we think of health benefits, and Christine Verbeek later on will go into more depth into the mechanism or potential mechanisms, and Mariuka will show the evidence that is already present. But I would like to go with you through some of the things that fermentation can do to foods, which can then be linked to health effects. Transformation of food constituents is a first element. Food texture affecting processes. Second, synthesis of bioactive compounds, of course, and then provision of live microorganisms in food is a fourth element that I want to touch upon. And if we look at plant-based foods, very often we are talking about foods or raw materials that are rich in starch, rich in protein, rich in fiber as major constituents. We can also have minor constituents as phytate and others. Now, the fermentation processes are typically quite long. They very often reduce pHs, which means that endogenous enzyme systems are being activated. Next to that, also the metabolic activity of the microorganisms is present. So that combination of long fermentation times, endogenous enzymes that are activated, and microbial enzymes and microbial fermentation processes very often leads to changes in the molecular structure of those major constituents and minor constituents. And that is actually what we are also investigating in the HealthFerm project. What is changing? Why is it changing? And how does that affect health? Yeah. And so the changes can affect technofunctional properties of the constituents, can affect digestibility, can affect bioavailability of constituents for sure. And one example that you see here is the fermentation of black beans and kidney beans with Pleurotus austrateus, where there is a proteolytic enzyme system active. And you see that the fermented foods have a better protein digestibility compared to the control. So this is a simple example of increasing the digestibility of those materials and therefore having a better nutritional value for the product. The second example that often comes up is with regard to postprandial glycemic response. And there's a very nice review by Aurora et al., so the group from Marco Robetti and Raffaella Di Cagno in Bolzano in Italy, where they show the effect in a review of sourdough leavened bread versus yeast leavened bread with regard to glycemic index. And you see a lowering of the glycemic index based on the sourdough fermentation process. Now, there's another review from the people at Le Safre, so Léa Ribet and others, where they looked broader into some other aspects, and they said, well, if you look at sourdoughs, there is conflicting evidence, and there are sometimes nutritional benefits that are being reported, but often also no beneficial effects. So there is conflicting evidence, and one of the reasons for this is something we try to lay out in a review that we published very recently, where we looked at the different types of sourdough breads that are described in literature. And what you see there is you can call something a sourdough bread, but there's very often a very large variability in the properties of those breads, be it with regard to acidification. Yeah, we see pHs turning up from 3.3, but all the way to 6. We see that there's very different fermentation times, very different levels of sourdough addition or yeast addition. So that means that it's very difficult to talk about the health effect of a sourdough bread, because those sourdough breads are very often quite different in composition and production way of being produced. And that, I think, is a challenge that I will come back to later on, is that if we do nutritional trials, if we do intervention trials with humans, we have to make very clear that the products that we use are very well characterized. Otherwise, it is difficult to link a product to a health outcome. Phytate, I mentioned before, mineral bioavailability through degradation of phytate by phytases and alkaline phosphatate, releasing mineral ions is also of importance. Now, the second element is food texture, of course. We all know the importance of mastication and particle size of the food that we swallow with regard to digestion. If we have larger particle sizes, digestion will go slower. And so we know that fermentation processes will affect the food texture. And so there's also initial evidence in literature that different bread structures and different degrees of disintegration will lead to differences in digestibility. Going into the bioactive compounds, and that's also something that Christine for sure will go more in depth on. We know that if we have fermentations, there are different acids being formed. Lactic acid, quite known, acetic acid, propionic acid, maybe butyric acid as part of the short chain fatty acids, of which we know that they're very important in colonic fermentation, but we know much less of their importance in food fermentations and how these acids are absorbed in the digestive system when consumed and how that compares to colonic fermentation. Vitamin production. Here, a very small example of Estermophilus, where we see that different strains of the Estermophilus are able to produce very different levels of folic acid. Yeah? So we're talking about the same species, but different strains. Yeah? And so again, it is very difficult to talk about a specific strain, whereas we should be talking about our specific species, we should be talking about specific strains because they can differ quite a lot. Similar things for vitamin B12. It is important that if we reduce our animal-based products intake, that we compensate for the loss of vitamin B12. And that can also be done using microorganisms. Lastly, a very intriguing thing is about the provision of live microorganisms in food. We all know about probiotics, and we know that those probiotics lead to health benefits. But what is more and more surfacing today is the idea that the microorganisms that are present in the foods, not so much the prebiotic types, but microorganisms per se, will lead also to health benefits. And there was a study by Maria Marco and colleagues that was published not so long ago, where they looked into the NHANES database and looked into around 10,000 types of foods, looking at how much microorganisms were present on the different foods. And so they saw that the majority of what we eat are foods that contain virtually no microorganisms. So 96% of the food we consume is devoid of microorganisms. There are 3% of foods that have medium levels of microorganisms, so 10 to the 4th, 10 to the 7th colony-forming units. And then there's a very small percentage of the foods that we eat, 1%, where we have high levels of colony-forming units, 10 to the 7th and higher. And so if we eat 100 grams per day of those microbe-containing foods, then we have 10 to the 8th, 10 to the 9th microbes that we take in on a daily basis. And the question, of course, is there a correlation between the intake of those different food types and health outcomes? And the idea is, yes, it is. There is a relation there. And so there are associations that are digged up in this paper by Colin Hill and Mary Ellen Saunders of ISAP, where it is shown that there are correlations with systolic blood pressure, C-reactive proteins, plasma glucose, and so on. So I think we are at the start of this type of research where we try to link microorganisms and microbiomes to health outcomes. It's quite interesting. With this, I have come to the last part, which is 2 slides on future research in the field of food fermentation. And I think what we see is that the biodiversity of microorganisms today is not really exploited. We use very few microorganisms for fermentation. There's a lot more that we can do. And with the citizen science project of the HealthFerm project, we are also looking into increasing this biodiversity in fermentations. Engineering microbial communities, that I think will also become more and more important if we want to go from spontaneous fermentation processes to designed fermentation processes. We need to have that engineering of microbial communities. And as a last slide, understanding the health benefits of fermented foods and trying to boost health benefits of fermented foods. But that means that we need very well controlled, randomized controlled trials with thoroughly characterized products. Again, I showed you the example of sourdough bread, where we have very different properties of those breads. We need to make sure we characterize them well enough to make sure to make a link between the product that we eat and the health outcome that we have. There's also a lot to be gained from, let's say, open access. So EU is focusing a lot on open access of data, and there is an incredible amount of data available out there, or should be available, where if we can gather all that information and do the right bioinformatics analysis on it, we can probably already derive a lot of information from what is already out there. I think the study of Marco on the NHANES database and of Marielle Sanders already shows that. With this, I would like to thank you all for listening, and I hope I've set the scene for the presentation of Christine and of Mariuka, looking into possible mechanisms on the one hand and existing evidence on the other hand. So thank you for listening, and I give the floor back to you, Jan.
Thank you very much, Christophe, for, I think it's a very nice overview of what HealthFerm is based on and what is the potential of fermentation technologies for future foods. The questions that were raised will be addressed after the presentation. So I'm going to introduce Professor Christine Verbeke for the next presentation. She's a full professor, head of the Laboratory for Digestion and Absorption, also at the Catholic University in Belgium, and her research, her research focuses on the bacterial metabolisms in the human colon, and she's particularly interested in how diets affects the bacterial metabolism, and most importantly, what are the consequences for the host health. And she uses a lot of modern technologies to look at the potential mechanisms. Christine, floor is yours.
Okay, so I hope that you can hear me right now. Thank you very much, Jan, for this kind introduction. Also from my side, welcome to this webinar. And so if you would enter in Google the search terms fermented foods and health benefits, you will find about 26 million hits and plenty of reasons why you should eat fermented foods. Now today, I will not talk about the health benefits themselves, nor about the evidence that we have for those benefits, because that's the topic of the talk of Mariuka. And instead, I will focus in my presentation on the mechanisms that might explain the potential health benefits of fermented foods. And for that, I will limit myself to plant-based fermented foods rather than also including dairy products like yogurt and cheese, because plant-based fermented foods is what we study in the HealthFerm project. And as you already heard from Christophe, a first mechanism for health benefits is the fact that fermented food products may be a source of beneficial bacteria. And so by consuming fermented foods, you may considerably increase the numbers of live microbes in your diet, and estimates indicate that it can be up to 1,000-fold. Interestingly, several of the microbes that are found in fermented products are similar or even identical to known probiotic species. And you can see here some examples of lactobacillus species with recognized probiotic activity that also have been found in fermented food products. However, not all fermented food products contain viable microbes at the time of consumption, and this mainly depends on the type of processing. For instance, sourdough is produced by a consortium of lactobacillus species and a yeast, but when you bake your sourdough bread, all your microorganisms are killed. So when you consume sourdough bread, there are no viable microorganisms anymore. This table provides some examples of fermented food products from different regions in the world and the microorganisms that they contain. And as you can see here in the last column, also their concentrations are in an order of magnitude, varying between 10 to the 6th and 10 to the 10th per gram or per ml, which is quite considerable. Now, for those microbes to have an effect on health and via modulation of the microbiota, they should be able to affect the microbiota composition in the large intestine. And I show you here a study as an example in which females consumed kimchi either in a low dose of 15 grams per day or in a 10 times higher dose of 150 grams per day for 7 days. And after that, the fecal samples were analyzed with 16S or RNA sequencing. And here then you see that high doses of kimchi reduced a number of potentially harmful genera like Listeria, Clostridium, Enterobacter, Prevotella, and Shigella, whereas there was not really an effect on the potentially beneficial microbes. There is here, you see an increase in lactobacillus species, but that was not statistically significant. Now, secondly, food fermentation goes along with the production of a range of bioactive compounds. And the first category of such compounds are the polyphenols. A number of plant-derived foods, such as black tea, coffee, dark chocolate, but also blueberries, apples, and vegetables like broccoli, spinach, and onions, and asparagus, are rich in polyphenols. So the polyphenols are classified based on their chemical structure, which can be very variable, as you can see here. And it is also important to realize that within the plants, the polyphenols are often decorated with monosaccharides, which may reduce their biological activity. Now, several of the microorganisms that are used in food fermentation are able to modify these polyphenols because they secrete enzymes like tanases, esterases, decarboxylases, and glucosidases. And in general, we can say that the larger molecules are usually degraded to smaller ones. Glycosidic bonds are often hydrolyzed to generate the agglicons. And then there is also a huge number of biotransformation processes that is also involved in the formation of flavor and aroma. I show you here just as an example the possible biotransformation starting from coumaric acid or ferulic acid. But if you would like to have a more extensive overview of all these biotransformation processes, I would refer you to this reference here, in an extensive review in the September issue of Foods of 2023. The opposite also holds true. So the polyphenols in the diet also affect the microbiota composition. And I show you here a study in mice. So the control group received just a standard diet. The positive control received a high-fat diet. Then there was a group that received unfermented celery juice in addition to the high-fat diet. And then there were 3 groups that received increasing amounts of fermented celery juice, also in addition to the high-fat diet. Now, the fermented celery juice differed from the unfermented one in the sense that it contains more acids and more total phenols, also more flavonoids, so more polyphenolic compounds, also a bit more vitamin C and superoxide dismutase. And then what they saw is that addition of the high-fat diet reduced the biodiversity, the diversity of the microbiota in those mice. Addition of the unfermented celery juice could not prevent that, but addition of the fermented celery juice prevented in a dose-dependent way this reduction in diversity. Here in the PCA plot, you also see that the samples from the animals that received the highest dose of fermented celery juice resemble best the ones of the animals that on the standard diet. Also an indication that the diversity reduction was prevented. At the phylum level, you will see that the high-fat diet increased the proportion of Firmicutes at the expense of the proportion of Bacteroidetes. That's what we expected, that's what we know happens upon a high-fat diet. That is also then reflected in a much higher Firmicutes to Bacteroidetes ratio. And then you see again that the fermented celery juice, addition of this fermented celery juice in a dose-dependent way could revert this change and bring the proportion of Firmicutes to Bacteroidetes almost back to the proportion that we see in animals that get the standard feed. And also on the level of the genus, so the genera, you can see that the fermented celery juice increased a number of genera that we consider beneficial, such as the Ruminococcus, Lactobacillus, and Faecalibacterium, whereas a number of other genera were decreased after addition of the fermented celery juice. So it's clear that these polyphenols affected the microorganism in this study. Now, in addition to antioxidant activity, polyphenols are also thought to protect against cardiovascular disease, diabetes, some cancer, and some neurodegenerative disorders. But it is important to realize that these effects have mainly been studied in vitro and animal setups, and that it is important to take also some complicated elements into account when trying to transfer or translate these results to human health. Complicating elements such as matrix effects, enzymatic interactions, reactions with other foods, but also host characteristics like the gender may play an effect. Also, genetics of the host can have an effect on the biological activity of the polyphenols. It's also important to have knowledge on the pharmacokinetics of the phenols, because that is essential to be able to estimate a correct dose for in vitro studies. And also the fact that the polyphenols are extensively metabolized by the microbiota is important, because sometimes the metabolites have more pronounced biological activity than the original mother compound. So you need to know if you have to study the metabolites as well, or if you only have to study the mother compounds. And then finally, it's important that results of isolated compounds are always compared to that of the actual available food source. So taking actually this matrix effects into account. A second category of bioactive compounds are the bioactive peptides. And so bioactive peptides are typically made of 2 to 20 amino acids that can be directly extracted from fermented foods. Depending on their amino acid composition and sequence, they can have a variety of biological properties such as antihypertensive, antioxidant, antibacterial, anti-diabetic, immune modulatory, and anti-inflammatory and anti-cancer effects. And so far, more than 4,000 of these bioactive peptides are known and have been characterized, and you can find them in this freely accessible database. So you have here the link, and then you have to push this button where you can find a list of all these known bioactive peptides. An important remark is also that bioactive peptides can, unlike macromolecular proteins, reach the systemic circulation via endocytosis or intercellular transport or diffusion. So there are several mechanisms by which they can reach the systemic circulation. In this table, you see a number of peptides that are able to inhibit the angiotensin converting enzyme. This enzyme converts angiotensin 1 to angiotensin 2, which is also the target of several antihypertensive drugs. Angiotensin 2 is a vasoconstrictor, which increases blood pressure. So if you can inhibit its formation, your blood pressure will reduce. And such peptides have been isolated from different sources, and their amino acid sequence has been determined, and you can also see their activity. And it's also clear that this activity can differ very much and really depends on the amino acid sequence, but it's very hard to predict based on the amino acid sequence what the efficiency will be or what the type of activity will be. Other peptides, for instance, have shown varying levels of antioxidant capacity. Here again, you see that there's a big difference in the sequence of these peptides, also in the length of these peptides and in the biological activity. So again, it's very hard to predict the activity based on the amino acid sequence. And then there is also a number of peptides that has antimicrobial activity, with again the same comments, very different amino acid sequence and very different biological activities. Then also the production of short-chain fatty acids from the fibers in these plant-based products is something that can affect human health. So short-chain fatty acids are mainly acetic acid, propionic acid, and butyric acids. And in physiological conditions, they are produced in the large intestine by bacterial fermentation of dietary fiber. But when you ferment your food, your fiber-rich product in advance, so before you consume it, you can generate those short-chain fatty acids already in the product itself. And we think that these short-chain fatty acids may be the link between fiber consumption, the microbiota, and the host, and especially the health benefits that fiber confers into the host. Here you see an overview, a table of a selection of fermented food products and the amount of short-chain fatty acids that they contain expressed per serving. then it's immediately clear that these fermented foods mainly contain acetate and much less propionate and butyrate. It's only here the gherkins and pickled onions that also contain some of those short-chain fatty acids, but it's mainly kombucha and here vinegar that contain the highest amounts of short-chain fatty acids. Now, short-chain fatty acids can have local effects in the gut via activation of G protein-coupled receptors here on the enteroendocrine L cells in the gastrointestinal tract. Activation of these receptors results in the release of gut hormones GLP-1 and PYY. In addition, short-chain fatty acids may also reach, at least partly, the systemic circulation, and in that way they can also affect organs on a distance, such as, for instance, adipose tissue, the liver, the pancreas, or eventually the brain. And we hypothesize that the effects of short-chain fatty acids may differ when they are absorbed in the small intestine, which happens when you consume fermented foods where the short-chain fatty acids are already present, compared to when they are absorbed in the large intestine, which happens when you consume dietary fiber, for instance. And the reason why we think that is that in contrast to colonocytes that consume short-chain fatty acids and convert them into ATP to generate energy, so they use them as an energy substrate. Small intestinal cells don't do that. They use glucose or glutamate as energy source, and therefore we assume or we hypothesize that the systemic availability, so the fraction that reaches the systemic circulation and may have an effect here on other organs, will be much higher when you administer short-chain fatty acids or when you consume fermented foods compared to administration of short-chain fatty acids in the colon. In contrast, because the density of these enteroendocrine L cells is higher towards more distal parts of the gastrointestinal tract, we think that the effect on gut hormone release will be slower or lower when you administer short-chain fatty acids into the small intestine compared to the colon. And that is exactly one of the hypotheses that we are testing right now within the HealthFerm project. Okay, two more mechanisms by which fermented foods may induce health benefits is by increasing bioavailability of nutrients and by reducing compounds with negative effects. And I will discuss these together because they are related to each other. It's mainly by reducing the compounds with negative effects that you can increase the bioavailability of nutrients. Fermentation of plant-based foods reduces the amount of tannins and of phytates, which can be considered as anti-nutritional compounds because they complex the minerals and prevent them from being absorbed. So reducing these compounds results in a higher bioavailability of mainly iron and also zinc, despite the fact that the total amount of minerals is often somewhat lower in the fermented food products. The impact of fermentation on vitamins is a bit more variable because we see that, for instance, the amount of vitamin C decreases in almost all vegetable fermentations with increasing fermentation time, but in contrast, B vitamins generally increase during fermentation, and also vitamin K can be produced by some strains such as Bacillus subtilis. And then a final mechanism that Christophe already mentioned is that fermentation improves the digestion of nutrients and especially protein digestibility. So compared to animal protein, which has a digestibility of about 90 to 95%, digestibility of plant proteins is generally a bit lower and varies between 70 and 85%. And fermenting plant-based foods can be a way to increase their protein digestibility. Here you see an example of pea protein concentrate that was fermented with Lactobacillus plantarum up to 11 hours, and you see that then gradually the total phenols and the tannins are increased or released, and that at the same time chymotrypsin and trypsin inhibitory activity is decreased, which results in a slight increase in protein digestibility. It's not really spectacular, but there is a clear increase in protein digestibility. An important remark here in this study is that the protein quality decreased with increasing fermentation time, and that seemed to be because this specific Lactobacillus plantarum that was used for the fermentation consumed the sulfur-containing amino acids. And sulfur-containing amino acids are essential amino acids, so if you reduce them from the food, the protein quality decreases. So when you ferment food products to increase their protein digestibility, it's also important to choose a strain that does preferably not consume itself the essential amino acids. Another example is here looked at the effect of two cultivars of chickpeas and also faba beans. And here you see the protein digestibility when they were raw, of the raw products, after natural fermentation and after fermentation with lactobacillus. And depending on the cultivar and the type of fermentation, you can see that you can modify the protein digestibility, especially for the chickpeas and maybe to a lesser extent for the faba beans. So this brings me to the take-home messages of this presentation, where you can say that mechanistic studies in cell models and laboratory animals identified potential mechanisms for health benefits of fermented foods. These mechanisms are definitely not uniform for all fermented foods, but depend very much on the type of the food, the type of the microorganisms, and the type of processing. And what is also important is that those mechanisms in itself should not be considered as a health benefit. For instance, showing a reduction of phytate, which increases the bioavailability of iron, that itself is not a health benefit. It's only a health benefit when you can also show in humans that it will result in, for instance, a better bone mineral density, so that it really has a beneficial effect on the humans itself. So all the benefits that can be mediated by these mechanisms should be also confirmed in human intervention trials. I think that is really a very important aspect and a message that I would like you to take home. And with that, I would like to thank you for listening, for your attention. I'm happy to answer your questions at the end of the session.
Thank you very much, Christine. It's a very nice overview of what is feasible, what we think is feasible with fermented foods in relation to health benefits. I think we can continue now what is actually already proven with respect to health benefits from fermented foods. And I really would like to introduce Marjukka Koleminen. She's a professor in nutrition at the Institute of Public Health and Clinical Nutrition at the University of Eastern Finland. And her expertise is within health effects of diets, foods, food components, and structures that she has studied in acute, short and long-term interventions already over 20 years. Marjukka, floor is yours to explain what is already known about health benefits of fermented food intake.
Thank you very much, Jan, for the kind introduction. I will show or share my screen now. I hope you can see it now.
I'm not seeing your screen yet. Yeah, we see your screen. Thank you.
Okay, thank you. Okay, I will run through with you a little bit of glimpses of the evidence that is there in the literature at the moment regarding the fermented foods. My main focus will be on plant-based fermented foods. If I'm able to, okay, now it goes on. So first I would like to show you some results or convince you that why plant-based foods, and then go to the more to the fermentation. Of course, we know already for a long time that there are several health benefits of the plant-based diets. And I mean not vegan diets, but also mixed, but more focused on plant-based foods, like how plant-based foods compose diets. But within, let's say, within the last decade, we have learned a lot about the whole the plant-based foods and how they interact with guts, and how gut-mediated functions are really evident for our health. Plant-based food, they contain complex food matrices, so as already Christoph and Kristin told you, and they have two-sided effects, health effects. They might reduce the nutrient availability, which can be a health benefit, but it also might be unbeneficial in terms of some like a mineral bioavailability. We will go more deeply in a second. Dietary fiber, we already know a lot about it, how it interacts with gut microbiota, what kind of elements it also mediates via the gut barrier function. It really seems to improve the barrier function by producing mucus, by decreasing the inflammation within the gut barrier wall, and also improving the gut barrier protein activities. They also contain a lot about the phytochemicals or bioactive compounds that we really don't know too much about, but plant-based foods are really a rich source of phytochemicals. And these all can mediate, all three compartments may introduce the effect via the guts and gut microbiota. And it might lead to decreased risk of chronic diseases. Here I would like to show you also how a couple of years ago there was this study by Desa et al. Where they really nicely showed that a fiber-rich diet really improves the gut barrier function. And today we really know that if we don't have so much fiber in our diet, it might lead to the so-called leaky guts, which is a name for the condition that lets or allows the microbiota produced unbeneficial metabolites to reach the circulation, and that way increase the risk of chronic diseases by producing metabolites in our like inflammatory factors or markers in our circulation. In addition, the fibers, as Kristin already told very deeply, also with the interaction with gut microbiota, they produce short-chain fatty acids, which also improves the gut barrier function, but also have a lot of other health beneficial functions. So these are like a sort of a basic elements that are nowadays known to be related with the plant-based foods. But why fermented foods? I would like to show you here a very concrete evidence on what fermentation does with the these plant-based compartments encapsulated nutrients. We did it together with actually current HealthFerm partners in Finland. This is called FibroFect study, and there we wanted to study really the whole grains, and we showed, as others already before, that whole grains contain a lot of dietary fiber, vitamins, minerals, unsaturated fatty acids, and biochemicals. And there we were able to show that the fermentation increases the bioavailability of proteins, micronutrients, and bioactive compounds using many different advanced methodologies. But these structures, although they are very beneficial in terms of mediating the health benefits via gut microbiota, they encapsulate these nutrients, as I already said. They slow digestion and absorption, and that can be causal for health benefits. It can be beneficial regarding glucose. It reduces glucose absorption and digestion, and that way keeps the postprandial glucose levels in circulation more stable. But it can be unbeneficial regarding the bioavailability of micronutrients. But here I would like to show you that this is concerning rye, and in native rye bran, you can see really that blue are the cell walls. So basically the fiber compartment, and encapsulated in these cells there is a lot of protein. And you can see that if you bake a bread from this kind of that includes this native rye bran, it's still in the bread. It's mostly in the similar kind of condition, which makes it really concrete to understand that this might decrease the protein bioavailability, for example, because our digestive system is not very efficient in breaking this kind of structures. When you then process the native rye bran with the enzyme and fermentation, you can see that in every step, you can see that these compartments, fiber compartments in the rye bran, they start to break. And here in the final product as a bread, you can really see that these compartments are broken down and release of the nutrients is happening. So this is really like making it very, very concrete. What are the sort of like a hypothesis from the nutrition point of view, why we expect the fermented foods to be more beneficial also in nutritional terms. Well, fermented foods, Christoph already showed you this, but I would like to address that also this statement, because it has also, in addition that it describes very well that what are the fermented foods and so on, it also gives a nice overview of the health related evidence. And it comes very, very clear also while reading this statement is that yes, there is some evidence related, epidemiological evidence that is based on the association. So fermented foods seem to be lowering risk or decreasing risk of chronic diseases, but there are only few well-controlled randomized trials to study the fermented foods. And the main evidence is really on fermented dairy. And when you go to the plant-based fermented foods, the evidence is really, really varying, really scarce, and that is the reason why we need much more evidence, well-designed studies. And actually this we will do within the HealthFerm project. I would like to show you a quite recent study where the Vastyuk et al. Were comparing high fiber diets to high fermented food diets. This is, I would say, one of the first studies that it has really nicely designed study, and it's really using a lot of different kind of omics and putting them together and to show how these different, especially fermentation, is having added value in regards of health benefits. So they divided their study population into high fiber diet that included various fiber sources, and then high fermented foods diet that included various fermented products. And of note is that there is also dairy, so it's not only plant-based foods that were fermented in here. But they could really show also, although it's quite a small population, but by using these advanced methodologies, they could really show that these diets, they produce quite different effects, especially in the gut microbiota. So these diets, they had a beneficial effects. They increased the gut microbiota, but there were different microbes where their abundance were increased differently in these two diets. And what was also shown was that this high fiber diet had the same benefits that has been shown before, but there was much more variation in, for example, in the inflammatory responses, as they say here, that the micro functions were tracked with the inflammatory functions, but they were more personalized. So depending on the individual, the immune responses were sometimes even increased, but most of the population showed decreased immune responses. But fermented food diet increased really the overall microbiome diversity and decreased the markers of inflammation. So this shows that fermented foods might have added value when comparing to quite similar diet made in plant-based foods. Well, and when we go really to the plant-based fermented foods, as I said, it's really, the evidence is really varying. And I hope that I can also show you the challenges of doing dietary interventions. So first of all, or comparing the results of the dietary interventions, because there are so many different plant-based foods that are traditionally used as fermented. So it's really hard to find really strong conclusions, really strong evidence that you could say that this is always happening with plant-based fermented foods in terms of the health impact in humans. And yet still the most of the evidence, both epidemiological and clinical trials, is based on the fermented dairy. So this is also something that is really evident when we go through the literature. So a couple of examples here. So as we know, gastrointestinal tract is a major component of human immune system. So it has been estimated that some 70% of human immune system is located in the gastrointestinal tract, or it is mediated, its function mediates the immune system. Then it's very evident that food may have a really strong impact on the immune system. This meta-analysis uses 26 randomized control trials with a quite nice number of population. And the authors say that fermented foods consumption is favorably affecting to the TNF-alpha levels, but no evidence in sensitive CRP or IL-6 as inflammation markers. Here you can see the figure on the IL-6 where the meta-analysis have been done, and yet, yes, there is not effect or added value with fermented foods. And here is TNF-alpha, so you can see that there is very, very slight slight improvement in TNF-alpha markers. Unfortunately, these studies were regarded not in very good quality, so also this calls upon the new studies to be done. And kombucha and kimchi are also familiar in Western countries nowadays. So it is a little bit surprising that most of the evidence from kombucha is really based on animal models. But kimchi consumption in healthy volunteers seem to improve many of the cardiometabolic risk markers, like LDL cholesterol, HDL cholesterol, other lipids, some cytokines. So it seems to be related to decrease of risk markers of cardiovascular diseases. This is a meta-analysis on how fermented foods have an impact on glycemic control in diabetic or pre-diabetic populations. The studies are really varying here. They are from 2 weeks to 12 weeks. They all are RCTs, so randomized controlled trials. But again, most of them are made in dairy, and there are only few Asian-based fermented plant-based food studies. And you can see here that again, a slight improvement in glycemic control found in these studies. This is a quite interesting study, and there they wanted to study that if the live microbe, including sauerkraut, would have added benefits compared to fermented sauerkraut. So both groups within these IBS patients, so patients had irritable bowel syndrome, and they were randomized into 2 groups, either consuming sauerkraut or then consuming sauerkraut that had or included these live microbes. But there were no added value. Both diets seem to show a little benefit, but not added benefit while there were live microbes in the diet. This is an example on the sourdough bread and GI health. So there are many different studies here, but the main issue was that these studies were investigating the impact of sourdough baking on nutritional content of breads and their potential to lower the content of non-digestible oligosaccharides. Some of these studies found that there were lower glycemic response, and they improved the GI symptoms that these volunteers in these studies were experiencing. But there were no difference in the markers of inflammation compared to or other products that were consumed in these studies. Regarding the rye, we have of course done a lot of studies in rye, and also the colleagues in Sweden and in Denmark have done a lot of studies in rye. And rye is many times traditionally consumed as a fermented bread. And it seems to have a lot of different health outcomes. It can affect via its structure, which is really tight, texture, its dietary fiber content, and many bioactive compounds have been related to satiety, insulin economy, glucose metabolism, improved inflammation, and improved lipid metabolism. And it has been, because these interventions are, of course, done with in risk markers, they are assumed to also be related to decreased risk of chronic diseases. But what is remarkable and what is still sort of not very well known why this happens is that this traditional sourdough rye bread, it increases glucose to the same level as wheat bread after a meal. But somehow the insulin levels remain to be lower compared to those after eating wheat bread. So why insulin remains lower when the glucose levels after a meal are similar? This is something that has not really been shown what is the cause of it. It's not related to incretins. It might be related to gastric emptying or consequently some other hormones, but there is no really evidence on what is ongoing. It has been nominated as a risk factor, which might be fiber, it might be bioactive compounds, it might be matrix, and all of them together. So this is something that is really thought that it comes via the fermented rye bread. I would like also to show you the one that is really interesting, one of the gut-mediated health outcomes from the fermented foods. It seems that in preclinical studies, so with animal models, the consumption of fermented foods seems to be relieving depression and anxiety. In humans, the literature is again really sparse and heterogeneous, and those studies have significant limitations. But you can understand how complex dietary interventions are to be done within this kind of phenomena. But we need again much more studies in this. But there are many sort of like provisional or hypothetical mechanisms that could really be linked with the relief of depression, anxiety, keeping in mind what we know on gut-brain axis at the moment. Last of the glimpses that I have gone through here is one of our studies. This is actually the same that I showed you the microscopic pictures of the fermented foods. Is a comparison between the whole grain rye bread, which is traditional fermented rye bread, and this group is consuming this bioprocessed rye bran baked into the white wheat bread. And this is a crossover design, so all the volunteers were eating or both breads as a part of their diet. What is peculiar that after these diet periods, we can see in the meal study, in a standardized meal study, we could show that there is a slight difference in a postprandial production of short-chain fatty acids, which is a little bit peculiar because short-chain fatty acids, their production in the blood circulation after eating a standard meal we assume that it comes later when the food is getting into the intestine. But it seems that it's something that is reflected by the diet already before, instead of really only the postprandial effect. So here we can say that this rye included diets, especially the whole grain rye, the traditional fermented one, had the impact on gut microbiota that was reflected in these responses. But what was really interesting in this study was that we did, together with Professor Kati Hanhineva, metabolomics analysis in serum samples on these volunteers. And what you can see is the decrease of serotonin every time they are consuming the traditional fermented rye bread. Increased serotonin levels in periphery. I'm not talking now about central system, but peripheral circulation. Increased serotonin levels have been associated with increased risk of chronic diseases. So we think that this decrease during the intervention with whole grain rye, traditional one, and decreasing of serotonin while people were eating that food is beneficial. We did then, because the 95% of the serotonin is really produced in gut barrier and enterochromaffin cells. So we wanted to make a examination in one animal study and could show that really the colon barrier production of serotonin was decreased with the high cereal fiber diet in these animals. So again, we can say that this kind of rye bread is modulating gut serotonin production that may really lead in long term the prevention of obesity, metabolic dysfunctions, maybe even decrease the risk of diabetes or cardiovascular diseases. Then to my conclusions, I have shown you a variety of different studies and meta-analysis. And as you can see, we can see probably slight benefits of fermentation of plant-based foods compared to control plant foods. So we really need to have more evidence on the actual health benefits and also the mechanisms how fermentation has an added value. But still the fermented foods, they may have their beneficial effect or induce their benefits by nutritive alterations of ingredients, as I show you, and also Christoph and Kristin have also showed you that it increases the bioavailability of nutrients. Some fermentation process may also induce the production of certain nutrients. It modulates the immune system via gut barrier function, via gut microbiota diversity. It has an impact on the presence of bioactive compounds, so also their bioavailability may increase. And this all has both effects on the intestine, but also in systemic functions. In HealthFerm, we are going to do 5 RCTs, studying the mechanisms and studying the mechanisms, studying the health effects, risk markers, but also doing a sort of overall dietary pattern study. So we are really looking forward what kind of results we will receive. Our own interest is really towards this tryptophan metabolism here at the University of Eastern Finland. So I would like to thank the group of researchers, talented researchers, that we are working here and producing new evidence. Thank you everyone for listening.
Thank you very much, Marjuka, and thank you for this very nice overview of what is already known with respect to health benefits, and maybe I'm short, but I can conclude actually that the major focus has been on dairy foods, and there is not that much known about effects of plant-based fermented foods. And still, we consume probably more of energy by plant-based fermented foods than by dairy foods. That's peculiar, actually. Before we start addressing the questions, I really would like to address also an announcement in that sense, that HealthFerm will also have a symposium at the conference in Nantes, the ICBC 24 conference, which will be organized in Nantes from the 22nd to the 25th of April. And I think it's very valuable to be there as well in Nantes. Having said that, there have been posed several questions from participants, and I think I'm going backwards through the presentations. One of the questions is, amongst others, about, okay, there is a overwhelming positive view of fermented foods in the public arena, let's call it that way. How is it possible that there is so few studies available, except then from the dairy area, with respect to the health benefits of fermented foods in general on plant-based foods? Or so what is happening? Why is the public so interested, and why is, maybe you can say, why is science lagging behind? Who wants to answer this question? Marjuka.
I can start, and for sure Kristin and Christoph can add their views, but I can say that this kind of human studies, they are really complicated to be done, and there are so many various products that we should study. So it's really, if you think the traditional fermented foods, there is vast number of those. So it's really like you take a bits and pieces of these foods and make a study. Of course, researchers want to do the study from their own points of interest. So that's why it seems like it's very scattered. We can put the fermented dairy in one pot, so it's yogurt or sour milk. So it's so much easier compared to this vast variety of plant-based fermented foods. And as I said, it's really hard to make a well-designed human studies when you have how to control them so that it's really showing the additional value of fermentation as such, and what is the target population that relates to generalizability. So there are so many things that I can tell you that it is a little bit challenging.
Can I also put the question on the table? Is it also about the money of the industry that is behind it?
Behind what, Jan?
The dairy industry, of course, has a global lobby and a global organization, and plant-based foods do not have a global organization as such. So how does that influence the science behind it?
That's almost a political question, Jan.
Okay, we're not going to.
No. I think the EU is doing a very good job to support projects like HealthFerm to make sure that results are out there, irrespective of lobbying groups or whatever you have there. So if I see what they're funding in the area of fermented foods, it's quite interesting to see. So the microbiomes for sustainable foods is there, the project Domino, Cost Action Pimento, Wheat Biome, Tree Biome, and so on. So it shows, I think, the willingness of the EU and of governments to invest in this type of studies because they think it's important. So I think that's already a very good sign.
Mm-hmm. And about the translation from animal studies to human studies, that is one of the last questions that was posed. And there is of course a lot of studies available from feed. What is the effect from feed on the growth, especially? But what can we learn from, for instance, the monogastric animals like pigs, and to extrapolate that to the humans instead of doing all the human studies?
Go ahead, Jesse.
Yeah, shall I say something? Well, I think for that pigs are good models. Their gastrointestinal tract is very similar to that of humans in terms of anatomy, physiology, immunology, nutrition, and so on. But in the end, you want to have an effect in humans. So I think if we should not do big trials if we can do it in humans. We should do big trials for the aspects that we cannot test in humans. Because in the end, you want the evidence in humans. But some things are not feasible to test in humans. For instance, you cannot take that much tissue samples and liver biopsies and whatever kind of things. And so for mechanistic aspects, you can use the animal models. But to demonstrate the health effects, I think you need human studies.
Mm-hmm. And if you, yeah, Christophe?
No, I completely agree. So if you want to look to humans, you have to look to humans in the end. That being said, I think if you look at broiler production, for example, a broiler can be a very good model. The same principles very often apply for what we see in humans. But on the other hand, whatever you do on broilers is also useful for broilers, in the sense that by studying it, you can improve well-being of the animals, you can improve possibly performance. But a lot of focus is going today to animal well-being, so footpath lesions, avoidance, that sort of thing. So we should use the right studies for the right things, and that's one of the reasons why in HealthFerm we do not have animal models. We just we do the experiments or the intervention trials with humans immediately.
Yeah. In that respect, one of the questions is also about the magnitude of the effect. When do we call something a health benefit, and how large should the effect be that you're studying, independent on maybe depending on the health benefit you are looking at, but is it feasible to do these studies? Okay, we have designed studies, of course, within HealthFerm to demonstrate potential effects, but how convincing are they? What is the actual health benefit that you can demonstrate with these studies in humans?
I think if you design human studies, you first have to choose a primary outcome, and you choose something that is clinically relevant. So you can, for instance, if you choose, I say, plasma cholesterol levels, for instance, that is important. And then you can decide, what do I define as a relevant endpoint? For instance, I want to see a reduction of 10% in plasma cholesterol. That is definitely clinically relevant, and then you design your study, and based on literature study and effect sizes, you can calculate a sample size that you would need to be able to show such an effect. So there are ways to design a clinical study in a proper way, so that you can, you have to base yourself on what is available in literature and what can we expect, and then define your study to be able to demonstrate it, and then you apply statistics, and if it is statistically significant, then you have demonstrated your health benefit. Of course, one study is not sufficient to have to make a claim or a health benefit, and you will need to repeat that in a different center with different people, maybe different population, to generalize it.
Mm-hmm.
Yes, I totally agree with Christine on this. And nowadays we of course also use other methodologies. We usually calculate the power of the study, so number of the volunteers based on clinically relevant markers. But we use also more advanced methodologies, different omics methodologies like a metabolomics or gut microbiome diversity, or what can we show in the microbiome. But there it is very hard to calculate the power based on those markers, because they are so variating, and there is no actually existing sort of like a standard that how much should one marker or marker profile change that you show that this is beneficial. And there we are then then we compare, and there comes the control. The control group is really important, because then we have a control where we compare the elements, that what kind of changes or modifications we can then show with these new kind of markers. And that I would say that adds a level of sensitivity, because sometimes these very traditional clinical markers, they are so well regulated, so it's sometimes quite hard with slight changes of diet to really show very dramatic effects, because that would mean a lot of unbenefits also with the control group. But then when you add a level of sensitivity by using this, you can really show that there is a slight modulation, because we also expect that diet will induce its effects in long-term effects. And sometimes we don't have a possibility to do such a long-term, like years of this kind of interventions.
Yeah, that's obviously probably the largest constraint that we face with nutrition trials. And just as a last question that we can address is, there seem to be many study clinical trials with conflicting unclear results of fermented foods versus the unfermented foods. How can the effects of live microbes and microbial products or microbial effects of the food itself be separated? So that is actually linking to your answer about nutrigenomics, metabolomics, and so on. And so is there a possibility to dissect these effects?
I think you can only do that if you have the right control product. If you have a control product that is fermented and the same product that is unfermented, and you can administer that and you see a difference, then it is due to the fermentation. So but that is of course very difficult to have, and you can try to have products with all, you can, for instance, I know, start thinking theoretically. If you would say, I have a fermented product that contains microbes and metabolites, then you could use an unfermented product, add the metabolites without the microbe and compare, and then it's due to the microbe, or you can do it differently. And just, but that's theoretical. We don't have all these separate control products. So in the end, we will probably not be able, but we do as good as possible and try to have control products that mimic as good as possible the fermented ones. And that's, I think, the only way to dissect that.
Yeah, I think they're pasteurized versus unpasteurized fermented products, and looking at kombuchas, for example, or others, that could also help in this regard, so that you look at one aspect at a time.
Metabolites, but not the microbes, and yeah.
There are several other detailed questions. I think it's already a 11:45, so yes. I propose to finalize our webinar, and I very much would like to thank Christophe, Kristin, and Marjuka for presenting their views on what foods we have, what are the potential mechanisms of fermented foods in relation to health benefits, and what's already proven, which I see there are quite some gaps with respect to the knowledge that we have, and that we definitely need to work upon, and that's one of the purposes, of course, of HealthFerm project. So, and I will also would like to thank the participants for their attendance and their patience, because we have run out of time, about 15 minutes. Thank you very much. The webinar is recorded. You will receive a link to the recording, so you can get back to the webinar once again and refresh your mind. Thank you very much, and have a very nice day.
Thank you, Jan.
Thank you.
Bye.
Bye.
Bye, everybody.